We're up to Unit 5 in our course examining key principles of brain health, or the biological basis of everyday problems like anxiety, fatigue, insomnia, brain fog, and mood problems. In this part we'll look at neurotransmitters, which are important brain signalling chemicals. You've probably heard of at least some neurotransmitters, such as serotonin or dopamine. And you could be forgiven if you've got the impression that they are the main determinants of mood and happiness and so on. And a lot of that perception is probably due to the fact that most psychiatric medications affect neurotransmitters. The reality is that they play some role, but they're only one relatively small part of the overall system of brain functioning. Let's recap where we've come from in the course. Although we're not specifically interested in medical problems, we're looking at a system called functional medicine as a way of addressing problems to do with brain performance. That means we're looking for underlying causal factors, and we're using lab tests to ask questions about functioning body systems so that we can find weak points in the system or places where we can apply our efforts to make the most difference. The strategy is based on the idea of the mind-body connection, that how you think and feel and act is underpinned by, but not necessarily determined by, your biology. Biochemistry you can think of in terms of raw materials. To work well, your brain needs to produce things like serotonin and dopamine, and for that you need a constant supply of raw materials. And biochemistry needs to be well regulated or controlled, meaning turned up or down according to need, so that you have just the right amount of, say, serotonin, not too much, not too little. So what are neurotransmitters? Well, if you've read books and articles about the brain, you probably already have at least a basic idea. They're chemicals used to transmit signals across a synapse, which is a junction between brain cells or neurons. So here comes an electrical signal heading in this direction down the axon of this neuron, and when it arrives at the synapse, it triggers secretion of neurotransmitter, which diffuses across to the neighbouring neuron, where it's detected by receptors, and thus affecting the behaviour of the receiving neuron. The first neuron then begins to reabsorb the neurotransmitter molecules for recycling via these transporter proteins, processes called reuptake. Reuptake happens for most neurotransmitters, but not all. Neurotransmitters can be excitatory, meaning they push the receiving neuron towards firing an output signal, or inhibitory, meaning they tend to block it from firing. Glutamate is the brain's main excitatory neurotransmitter, while GABA is the main inhibitory neurotransmitter. GABA tends to be calming, not just in terms of the firing of neurons, but also in terms of subjective experience. A lot of anti-anxiety medications tend to work by boosting the actions of GABA in the brain. Some neurotransmitters don't just work in this simple way across synaptic gaps, but they're secreted more widely and they diffuse out more widely. And rather than passing a specific signal between two neurons, they modulate the way that other neurons work and how messages are passed across normal synapses. This type of neurotransmitter are called neuromodulators. So here's a synapse, just like we saw before, and it's using maybe glutamate to send the signal across this gap. But here's our neuromodulator spreading diffusely and being detected by both these other neurons, and so changing the way that the standard signals are passed. Here are the best known neuromodulators, and they're associated with certain areas of brain function. Serotonin is associated with mood. Dopamine is involved in reward and motivation, also focus and attention. Noradrenaline plays a part in focus too, and general alertness. While acetylcholine has something to do with memory, and for example in Alzheimer's disease it's depleted. So the ones I've mentioned are just some of the best researched neurotransmitters, and there are many more besides. But from now on I'll just stick with the more general term of neurotransmitter rather than distinguishing between the two types. Now, an important point about neurotransmitter activity, or the size of the effect. And that's that it doesn't just depend on how much is released, but also how long it stays around. Earlier I mentioned reuptake, which is the process of recycling neurotransmitter molecules after they've been released into a synaptic space. Well, it seems that how quickly reuptake happens is the most dominant factor in determining how strong an effect a neurotransmitter has. So if reuptake is fast, then the neurotransmitter effect is lower because the neurotransmitter is not around for very long. And conversely, if it's slow, then you have a stronger effect. A lot of psychiatric medications are reuptake inhibitors, meaning they block the reuptake, so the neurotransmitter hangs around longer and so its effect is boosted. So I've mentioned there's a widespread idea that brain disorders, and especially emotional disorders, are caused by neurotransmitter imbalance. So for example, depression or mood problems are caused by lack of serotonin. But does it stand up as a theory? Well, probably the biggest reason for the popularity is that most psychiatric medications affect neurotransmitter balance. So for example, the most commonly used antidepressants are in a class called serotonin reuptake inhibitors, or SSRIs, and they block the reuptake of serotonin and thus boost its activity. Another class, the SNRIs, boost noradrenaline activity in addition to serotonin. But on the whole, the evidence for a causal link is considered to be pretty weak, not least because the evidence for the efficacy of antidepressants is rather limited. One of the strongest critiques in this area is this book by researcher Irving Kirsch. He examined all the studies of antidepressants, including unpublished ones, and he found that when you include the unpublished studies, then antidepressants don't appear to work any better than placebo. On the other hand, there is evidence that neurotransmitters play an important role in brain function. ADHD, or attention deficit hyperactivity disorder, is characterized by poor focus and concentration. And there's evidence that both dopamine and noradrenaline are important in these functions. One of the most commonly prescribed medications for ADHD is Ritalin, which boosts dopamine and also noradrenaline by blocking reuptake. When you take Ritalin, you can experience a fairly immediate boost in your concentration, and then it wears off a few hours later when the drug is cleared from the body. That's different from most antidepressants, which normally take a few weeks before you feel the effects. So with Ritalin, it's much harder to deny the connection. But the key point is that many other factors besides neurotransmitters can cause problems, and not all cases of depression are the same. And while low serotonin activity may play a role in some cases of depression, it's by no means all, and it may even be the case that sometimes there's excessive serotonin activity. It's the same with ADHD. ADHD and depression are really just names for groups of symptoms rather than specific diseases. And there are different subtypes, if you like, and for ADHD, boosting dopamine with medication is not going to work in all cases. So it's possible to predict some of the symptoms that follow from neurotransmitter imbalance, even if you can't say that those symptoms are always caused by that neurotransmitter imbalance. So, low GABA tends to lead to anxiety. Low serotonin can cause low mood or poor sleep. Low dopamine, low motivation and brain fog. Low noradrenaline, poor focus. Low acetylcholine, poor memory and focus. High glutamate can lead to anxiety and panic. High noradrenaline, also anxiety. So, to some extent, it's possible to use symptoms to make an educated guess about what neurotransmitters might be imbalanced. You've also got to consider what else might be going on, and also what might be causing the neurotransmitter imbalance in the first place. So neurotransmitter imbalance would be a mediator rather than a primary cause. So one possible cause for neurotransmitter imbalance is lack of supply of raw materials, and in particular amino acids. Amino acids derive from protein in our diet. A protein molecule is a long chain of amino acids that gets broken down by digestion, and the simple amino acids are absorbed and then used by the body to build new molecules, such as our own proteins or neurotransmitters. Most neurotransmitters either are amino acids or are chemically derived from them. For example, glutamate is an amino acid. Serotonin is derived from an amino acid called L-tryptophan. And dopamine is made from another amino acid called L-tyrosine. So again, problems with protein digestion could mean that you don't have sufficient amino acids absorbed, and that can lead to neurotransmitter imbalance. And in fact, you can use amino acid supplementation to address problems and improve symptoms. So for example, you can take L-tyrosine to boost dopamine activity and improve focus and concentration. It can be helpful, but not necessarily the best strategy, because to an extent you're not really addressing causes, at least not primary causes. Remember the three-level analysis of functional medicine. Neurotransmitter imbalance is at the middle level, that is, it's a mediator rather than a primary cause. So it could be a better strategy to look deeper for things like protein digestion problems. So let's just summarize some key practical points about neurotransmitters. Neurotransmitters do play a significant role in brain function, and imbalances can cause problems. But there's no easy way to measure neurotransmitter activity. You can measure neurotransmitters in blood and urine, and tests are available commercially, but the amount alone is not enough to determine activity, because as we've seen, reuptake is such a significant factor. And secondly, neurotransmitters are used elsewhere in the body. So for example, you may have heard that most of the body's serotonin is actually made in the gut, and it's not going to access the brain from there. And so when you measure serotonin in urine, say, there's no way to isolate the brain component. To some extent, you can predict imbalances based on symptoms, but it's pretty limited because there's a lot of overlap and there are many other causal factors besides. Attempting to manipulate neurotransmitter levels using medications has pitfalls, because in the longer term the body can actually compensate for the increased activity that you've induced by down-regulating the receptors. And that may be true even if you use natural therapies like amino acids. Neurotransmitter imbalance is really a mediator of problems rather than the primary cause, and it may be a better strategy to target primary causes and then let the neurotransmitters look after themselves. So what else can cause neurotransmitter imbalance? Well, we've already mentioned nutrient deficiency, so amino acids like tryptophan, but also nutrients that can act as cofactors for the chemical transformations needed to produce neurotransmitter, and vitamin B6 is one of the key ones. Then we've got some of the usual suspects like cortisol problems. Earlier in the course we've seen that chronically high levels of the stress hormone cortisol are damaging to the brain and are associated with depression and can suppress serotonin activity by increasing reuptake, for example. In this course we're looking at biology as the foundation of brain health and emotional well-being, but the mind-body connection is two-way, and of course psychological and social factors such as loneliness can cause low mood and depression. Light exposure or lack of. It's known that serotonin activity is light dependent. So we've got seasonal affective disorder or SAD, which is depression experienced most strongly in the winter months. And it may be that low daylight exposure and the subsequent effect on serotonin is the mechanism for SAD. Toxins can affect neurotransmitter activity. I've listed a few here. Copper excess is known to increase levels of noradrenaline. Inflammation. We'll look at this in Unit 8. Methylation defects. Now this is a big topic and we'll need some explanation, so we'll turn to this next. The relevance of methylation comes out of work first done by Dr. Carl Pfeiffer and then later by the biochemist William Walsh. Pfeiffer began working with mental health disorders and especially schizophrenia using a nutritional approach called orthomolecular medicine. And he used lab tests on his patients. And after Pfeiffer's death, his work was continued by his collaborator, Dr. Walsh, who now runs the Walsh Institute, and they have a huge database of these lab test results. Pfeiffer and Walsh were looking to discern patterns in the results that they could use to predict the best course of therapy for patients. And they came up with a system of biotypes based on their observations. The biotypes are not specific to any particular disorder, but run right across diagnostic categories. Biotypes are defined in terms of biochemical mechanisms, and they predict responses to nutritional support. So for a biotype, some nutrients will help, while others can make problems worse. And two of the biotypes are based on methylation. We have undermethylation and overmethylation. Methylation is a fundamental biochemical process where a substance is transformed chemically by adding a group of atoms to it called a methyl group, which is a group of one carbon atom and 3 hydrogens, so represented as CH3. Methylation is a step in lots of different processes around the body. Some examples are neurotransmitter synthesis, detoxification, and also gene expression. Gene expression, remember, has to do with turning genes on and off, and attaching methyl groups around DNA is a key mechanism in turning genes off. And it's actually gene expression that defines the methylation biotypes. So to put it simply, in undermethylation, relatively more genes are turned on, and particularly those that control neurotransmitter reuptake. That means that in undermethylation there's more reuptake happening, and so neurotransmitter activity is reduced. In overmethylation it's the opposite. More genes are silenced, there's less reuptake, and neurotransmitter activity is boosted. Methylation problems affect principally dopamine and serotonin. So in terms of symptoms, we're talking about mood problems, motivation, focus, and concentration. So this is important because if you know you're an undermethylator, you can take steps to boost neurotransmitter activity. While if you're an overmethylator, there's a good chance that the standard range of therapies such as antidepressants, but including amino acid therapy, will actually make things worse. Instead, you've got to find ways to reduce neurotransmitter activity. So how do you know if you're an overmethylator or an undermethylator, or indeed somewhere in between? Well, to some extent you can judge it from symptoms, but it's very difficult to be sure, and really the best way is to use lab testing. According to Walsh, there are two options. Firstly, whole blood histamine, and secondly, the ratio between two substances, SAMe and SAH, and Walsh tells us that this latter one is the better one. Now, we're in danger of getting a little too technical here perhaps, but really the point that matters from a practical point of view is that these tests tell you in broad terms whether you're an over or an undermethylator. I'll just give you a little more detail. SAMe is S-adenosyl methionine, or SAMe as it's sometimes called. SAMe is the body's main methyl donor, and it's converted into SAH and then into homocysteine. Now, homocysteine is very useful to test because more or less independently of whether you're an over or an undermethylator, high levels of homocysteine correlate to lots of different chronic illnesses, including Alzheimer's and depression. So it's a powerful biomarker. Homocysteine can be reduced by supplementing specific nutrients such as vitamin B6 and folate. So we'll conclude by summarizing. Neurotransmitter imbalance affects brain function, but it's not the only thing to do so, and moreover imbalance is a mediator of problems rather than a primary cause. testing neurotransmitter activity within the brain is not really feasible. And in any case, manipulating neurotransmitter activity directly may not be the best idea in the longer term. A better strategy might be to look for and address primary causes, including defective methylation, which you can do effectively using specific nutrients if you know your methylation status.